that one had the light. That one did not have that heavier kappa.
Hey, where did you get the barcodes from? I
You
mean,
said-
from us, but
Yeah.
how did you isolate those?
So I go over here to my vloop file that I created for you all here, and I open it inside the vloop software.
I'm sorry, I can't see the VLOOP
I
file.
was about to open
Oh,
it. It just takes a minute here. Now let me copy this barcode. So I'm copying the barcode. I come down here to barcode. and-
Now, is that on our VLOOP?
Mhm. And so I pasted that in, and now here is this sequence for the heavy chain and the kappa chain, and the nucleotide is sequence and amino acids are right here. If you need the entire consensus sequence across the entire region, you can come up here and hover over this, and then you'll see that the sequence is listed right there, and you can just click on it and it will copy it, and you can paste it into your next Word browser, Excel, whatever browser that you want. But you have both the heavy and the kappa and their respective sequences here.
Now, what are those bars?
These are like mutations. They are indicative of like a mismatch. Green is your start codon, orange is mismatch. I've seen some purple deletions. The red should be a stop codon, and so forth. And if you click on these, you can zoom in to the actual sequence, and you'll then can come back to the color scale again and see, well, the orange is a mismatch.
Is there reference our sequence?
No, the reference is the reference from the
genome.
The genome, right?
So the consensus is consensus from our sequence. You know what
Correct.
it really
So
is.
if you had multiple multiple
Right.
barcodes listed here, it would then say, here's your consensus based off of all of those. But since there's just one barcode showing for this particular sample, that's the same. The consensus in the
But it's one barcode, but it's 5,000 sequences.
correct. Yes.
So it's over. So is there a way to see, all of the ones
Yes.
that match, and the few that I guess that were not?
Well, so if
Or
I
how
come
they
over
put that together.
here and I have come down to my view read support,
Oh, there you go. Okay.
and I've got to go find the BAM file, which I uploaded this morning for you all. So
good.
it's there.
All right.
So wherever it's located at in your all's area, that. And so let me make this a bigger screen. So
Did
that
it say upload BAM file? Is that?
it asked about where is the BAM file located
Oh,
at.
so-
And so I just went and pointed to where my BAM file is for it. Yours, I just uploaded this morning to Box into that folder that has the vloop file. And so now this shows the reads, all these little different colored rectangles with arrows
Oh, I see.
are the actual reads aligning to this sequence. That
And I see that it likes to read the 5 prime in.
Right.
Because of course that's the
Right.
closest. But so it doesn't have it doesn't have as much information about the area, especially the area we want, like the VDJ or something.
Well,
the DJ.
well, so
Well, J, I guess.
well, yeah, but you can scroll down and you'll see the other
Oh, okay.
reads that are aligning across the entire this particular clonotype. And these little lines in here are indicative of mutations.
I see.
And
So you got a mutation in that one,
but you don't
or
have it in
two.
anything else.
I mean, you got it in those two, but not.
Right.
So those they say, okay, that's not it.
I wouldn't consider that one to be real. As I was mentioning yesterday, if I looked at like just, I know this is not a good example here. There's nothing here, but let's see, like that one here. Let me pull up the purples.
Oh yeah.
Come here.
What does it mean?
That I still don't know. I think these are different clonotypes that are inside that. So like your 5,000 FASTQ files, these are the different cells that have this particular IG sequence. But
There
if I
are
look,
different cells,
I don't know. I'm not
because
sure.
it should just be one cell.
That's what I'm guessing. It's just one cell, but I don't know the color code yet. I haven't looked this up, and I didn't have a chance to look that up since yesterday either. But like here, you have a little mutation here at this, this little green mutation, but I don't see it in any of the other reads here, so I don't believe that it's to be real. I really don't see it in anything else either. It's just not.
Is it listed in any particular order of what do you call it? Fidelity or
No.
quality?
So most of this is already good quality alignments.
Oh,
So-
what do you see then?
Well, you can hover over any of these and click on the box, and it will then give you the mapping quality. So 255 is the highest score.
What does that mean?
It's just it's a numerical number. Like with the Fred scale and the FASTQ, it goes 0 to 40. In the mapping world, I wish that they stayed at 0 to 40, but they have certain tools have certain mapping qualities, and the highest in one is 255. Another one is there is no limit. The higher the number, the more it's better.
So 255 is good.
Right. It's mapped. Yes, perfectly.
How does it determine? How does it know? I mean, mapping quality means what? How does it
It took that read
Right.
and it aligned it to the VDJ sequences in the human genome, and this one mapped perfectly. There weren't any like insertions, deletions, or any problems. And when it gets a 255 score, it means that there's no other location that it could have aligned to as well.
no other location?
Right. So it's a unique.
What do you
map.
mean no other location?
So
in
a
the
lot of times you can have
genome.
multiple. Yeah, right.
Right.
In the genome. Right. So,
Yeah, because actually when we looked at what was it, 10x, we see like a few different light chains. Like it's trying to map.
mhm.
It says okay, maybe this one, maybe that one, kind
Right.
of thing.
But you can come in and click on any of these little rectangles here to get the information for the reason. There's plenty of information to read on. So
Anything we need to know, like
no,
I mean, but we don't know what it means.
if you're looking for mutations, this is sort of a place to quickly scan to see, well, is there anything that's common across all of them? And do you see a bunch of mutations like all these reds? Would they all be down here, or I'm only seeing in one or two reads, which maybe not as believable? Again, like I mentioned on the phone with you, mutations that are closer to the ends or the beginnings usually aren't as reliable as those that are in the middle, just because of sequencing
I see.
issues on the machine.
So those two in the middle there,
Yeah.
the red and the green thing
Mhm.
there, but they're only in that one,
Right, they're in these two reads alone, but they're not found in any of these right here.
or any of the other, like the blue reeds or green reeds, all those. It's really, or I don't know. I mean, there are mutations. You showed it in your overview.
Right.
How did it determine those are real mutations? If you go back to the big view.
Yeah, if I go back to
Yeah,
this view,
you
how does
say
it know
those-
that probably that it's got a. I don't know. I know how it works in other programs, but I don't know how it works in this one.
Can you go to the J region, because it looks like as you
Yeah.
go more down there, there's more mutations, right? Is that right?
Right.
I thought, or maybe a heavy chain would be better. So what's all that down there? Is it
So
like,
here's the kappa C. Nothing's out here. This is your J2.
so there's a lot of there's some.
Yeah, so like there's some deletions here, insertions.
Can you
mismatches.
zero in on? How do you zero in on that?
So I'm about to figure out if I click on that, it takes me there. So here's the junction between the kappa V 1D13 to the kappa J2, and so it's showing that this particular cell has a deletion at this spot. There are 4 base pair deletion. And then you see there's a mutation here. This little blue line here shows that there's an insertion of a TC at this location.
Oh, purple is deletion and
Yeah, I
orange
guess
is
so.
insertions.
Yeah, I have to always look at my color scale here. Yeah, that's right.
Oh, I see.
Yeah.
Okay.
So
Where's that color scale? I know you just
it's
did
the
it.
question mark.
Question mark, right.
Yeah, question mark will show you what the colors mean.
Okay. Now, is there a way to, I don't know, to convert that into, I mean, I know there is a way to convert it into a protein, but, then you say, well, this change here in the V region upstream, for example, change the entire, frame shift, and, it's just not going to work. I don't know. I don't know if there's a way to throw
I
those
believe
out.
There is. Let's see.
This can't be a real proteins kind of thing
Right.
based on something.
So let me see here. I just downloaded 2 files. I'll show you. I think it's this one.
And I think, by the way, I'm pretty sure we're going to talk to Greg Ippolito next Wednesday.
Okay.
And is that right? And I'm gonna Well, it'd be nice if you could come. It's
Yeah.
next Wednesday.
Send me the link or send me an email to remind me, and I can pull that up.
It's next Wednesday at what, 2 or 3? Okay, so if you're free, you're welcome to. In fact, you're definitely welcome to join us because you know what you're talking about. But what we want to do is now, what's the next step? We want to put these into his vector. We have his vectors now.
So,
okay.
I know that here I have the amino acid sequence of the CDR3 and of the nucleotide. So you have it both protein related and your nucleotides. And then for that one, here's the kappa sequence again. But
is there a way to? I mean, there is a way, but is there a way to tell productive versus non-productive ones?
You just need to search for the
Yeah.
stop codon.
You'd have to come back and search here. If I go back out, this is the start codon right here.
Mm-hmm.
And so it has ATG, so it should start all the way through. Now, is there a termination? It should be red. I don't see it in this instance. I see a bunch of, but the question is, is this software smart enough to see the deletion or an insertion or a mutation and know that changes the amino acid sequence? Majority of these tools that say, hey, here's your stop codon, are only looking for the actual stop codon. And they're not going, oh, by the way, upstream you had a frame shift where you lost a base and now you have a stop early. It doesn't, none of that does.
Well,
that.
there shouldn't be a stop codon there, right? I mean,
No, right. But I'm saying if one of these insertions or these mutations switched it out to a stop,
right,
then you would have an early termination. But the way I would, I guess, look at this is I would open this up to show that, and maybe copy this sequence, which I have right out here on the end, and then you can come in and look for the ATG and then just split it out, by threes and see if you find a stop anywhere along the way.
I mean, but
That's hardcore, but I don't.
oh no, we're going to do this. If we're going to do it for a few good, heavy and light chain genes. So we're going to study them in detail. But what program do you use? We used one before, and we found a lot more productive mutations in heavy and light chain from BD2 mice, right? Remember AID and all that was. So we did this, and we made a deal out of there's productive threefold increase in productive mutations in the autoimmune mouse. I mean, there was a lot of mutations, but the productive ones were increased. This is the old time, sequencing way. But
Right.
so we put, and I just forget what program we pasted it into to determine it.
It's just really manually.
No, I know. We only have a few sequences, just like before, so we can do it manually.
Remember, I just did it manually.
But what program did you use? Imgene or whatever, Impro?
I bet there's some tools out there that are been around for ages. I never convert stuff to amino acids.
Maybe we can
My
talk.
world,
Yeah.
Back in my proteomics day, I used to have to convert, but now I'm in genomics. I don't really care about the protein sometimes.
Maybe we can
So-
ask Greg about it. Because, basically, that we think that the highly mutated ones, especially in the diversity region, at least from our past or elsewhere, are the autoreactive ones. That's our model. And
Right.
that's how we're going to spot them and talk about them even. So we want to, but we don't want to have false, changes.
Right. And so I'm sure there's a way to do that. I just don't know. There's not in these tools for sure. So like I'm at the very front end of this, and so I pulled down the sequence, which is this up here. That sequence is this entire length of sequence here with the mutations in it included. And so you would just have to come over here, and here's the ATG, so GAC. So I'd have to come up here and look for it. I'd put it in Word or something easier to look for. But I'm sure there's ways of putting this in a tool and say, hey, find me the start codon, find me the end, and there's probably something. I just don't know.
Well, Greg would. Well, is the barcode missing? I mean, the barcode is not here, right? So
No, the barcode is not in this.
So there's other stuff like 5'
Right, so it would be the
sequences
5' sequence. So this is where the beginning of your what is it? The IGK V1D to 13 starts right here.
Oh, I see.
But these are also the reads that align to this, but they are also outside of the region. So it gives you a sense that yes, that aligned nicely.
Oh, yeah. And it started before.
Right, and it started before. And so you have, here's my start,
Right.
ATG. And so that way you can come in and take that entire sequence I copied and pasted and look for that ATG and then break it into threes or then copy it from that point on and put it into some. I bet there's a couple of online tools that convert things to proteins. I don't know them off the top of my head.
Is there a way to look at this? I mean, because this is at that amino acid level.
Mhm.
Let's say get the heavy chain and look at the D region, and just look at what the D. Look at the various because it does have a sequence.
Yeah.
But this
So-
thing is going to give us a few options, right?
Right. So this, gives you the entire V, and it tells you this is the V461. This is the actually we're looking at the kappa in this instance. So this is the V1D to 13, and then the next is the IGJK2.